Welcome back. Now, we all like to hear positive stories at the moment, and these next two speakers definitely have some positivity to share from their clinical pipelines. Our first speaker is Pascal Touchon, President and CEO of Atara Biotherapeutics. Atara have multiple T-cell immunotherapy product candidates in clinical development, including therapy for solid tumours, MS, and off-the-shelf allergenic platform. So, Pascal, over to you.
Hi everyone, pleasure to be joining this Advanced Therapy Connect today. And after our typical forward-looking statement, I would like to move on to what is really at the core of Atara activity, which is patient. We are really here to transform the lives of patients with serious disease through pioneering science, teamwork, and a commitment to excellence. Now, how are we doing that? We have developed a very differentiated and unique platform of activities that is really related to these 5 differentiating elements. We have a differentiated allogenic platform in terms of relying on EBV T cell, or Epstein-Barr virus T cell, that is fully scalable and allow us to have the right level of technology to scale up and deliver multiple cell therapy to patients. We have a very advanced, in fact, the most advanced allogeneic T-cell therapy product being developed in the US, Tab-cel, which is a first-in-kind late-stage oncology program for which we plan to complete our BLA in Q3 2021. And we are supported by very exciting clinical data there. We have also another asset in the clinic, ATA188, which is a potentially transformative treatment of multiple sclerosis. And this treatment is really, we believe, addressing one of the triggering factors of the progression of symptoms. And we have very encouraging data coming from our initial open study. And now we've moved into a randomized controlled trial. And last but not least, we have a next-gen allogeneic CAR-T portfolio validated recently by a strategic collaboration with Bayer that has just been announced a few weeks ago. And all that is supported by proven technical capabilities. All these aspects are really related to how we transforming cells from healthy human donors, either to address EBV-associated disease, it could be with Tab-cel, EBV-driven cancer, or with ATA188, MS, and maybe some other autoimmune disease. But we can also, on these EBV T cells that have been selected and activated to target EBV, we can insert a CAR, a chimeric antigen receptor, or another TCR, and that is leading then to engineered oncology product that can address different type of solid or liquid tumors there. So it's a very unique, very differentiated platform, but one that can be scaled up because we are leveraging all of our different pipeline products across that. Beyond this ability to scale up and leverage, we have really an amazing situation there on the next slide, where we see in fact, that we are able to develop fully with the idea to transcend the limitation of current cell therapies in harnessing the power of EBV T cells there. We are harnessing the power, and we have data showing today that this EBV T cell platform allows to have more persistence, durable efficacy, and very favorable safety profile, at the same time to be scaled up. And we have experience now in delivering this product within 3 days to hospitals across 3 continents there. So extremely advanced platform that allows us to deliver this allogeneic off-the-shelf cell therapy to many patients worldwide there. On the next slide, you will see that we have been able to develop a pipeline of product with, as I say, Tab-cel, which is now in phase 3 with the ALLELE study. And this phase 3 is supported by a phase 2 program that we have done both independently at Atara and with our partner at Memorial Sloan Kettering in New York, that has shown a high level of response rate in this PTLD patient with post-transplant lymphoproliferative disorders after previous therapy, where we've achieved response rate of 50% and higher, and very importantly, the ability to have durable response, bringing these patients to, in responders, to over 80% overall survival at 2 years, whereas these patients have usually a median life expectancy of just a few weeks or a few months there. So great progress for this patient with that type of therapy, and we are very hopeful that our phase 3 study will lead to similar type of result. We already did an interim analysis of that study that has shown that we have indeed demonstrated 50% objective response rate in this patient from independent oncologic and radiographic review. Now we have also now moving in phase 2 with AT188 or MS treatment. I'll say a few more words about it later on. And then we have a number of CAR T that are entering the clinic or in late preclinical stage. On the next slide, you can see that we have here all the support of very well-developed and near commercial, in fact, type of manufacturing platform with a dedicated expandable manufacturing facility in Southern California, and also a robust manufacturing process where very recently we've been able to show that we have now data enabling us to move manufacturing of this allogenic T cell into stirred tank bioreactor, allowing to have a much higher yield, a much higher efficiency of manufacturing. And that, according to a plan, will allow us at a later stage to have a cost of goods manufactured very close to any type of biologic like monoclonal antibodies. So great progress on having a very advanced manufacturing platform, but also technologies that allow us to move this cell therapy into any biologic type of typical cost of goods manufactured. On the next slide, you will see that we have these 3 strategic priorities at Atara. Tab-cel, we already discussed it, in different indication addressing unmet medical need in various ultra-rare disease. So it's really like a pipeline in a product type of situation with tab-cel across different type of EBV-driven cancer. And then with AT188, as I said, this is really potentially a transformation of MS treatment. If indeed the randomized controlled trial that we're pursuing is confirming what we've seen in an open study, which is a possibility to reverse disease progression in some patient with an improvement on disability, which has not been shown, and no treatment today approved in progressive MS has ever shown that. So it's a real big game changer that we're developing then in MS. Now, moving on the next slide to our third priority in bit more in detail, which is around our platform to do a next-gen allogenic CAR T. What we're doing there is really, as I said, to leverage the properties of these EBV T cells and adding with next-gen technology unique features in the design that allows to have an allogenic CAR T. That could be around a multi-targeted CAR with an AND gate or an OR gate to be able to address specificity of some disease and with good tolerability or even higher efficacy, and also to use new co-stimulatory domain that are really have been developed to lead to less T cell exhaustion and hence more functional persistence of the cells. And we've recently demonstrated in preclinical studies that we indeed achieve that longer functional persistence, allowing the T cells to be able to expand and eradicate tumors even after several stimulation by the antigens there. And we also have built in this design, especially for solid tumors, a PD-1 dominant negative receptor. And that's very exciting because that's a way to try to have an intrinsic checkpoint blockade that will unlock the solid tumors, the CAR T in the solid tumors, and unlock the tumor immunosuppressive microenvironment there, and allowing to have this activity will also lead to a longer persistence and the ability to eradicate tumors after several tumor re-challenge. So all of this means that we're optimizing the design of this type of allogenic CAR T to really address the need of specifically some liquid tumors and solid tumors. On the next slide, you will see that this has led us with this significant advance in our platform to be able to start a strategic collaboration with Bayer, and we're very excited about it, around mesothelin CAR T, not only because this is a recognition of Atara Biotherapeutics advance in the field of allogenic CAR T, especially for solid tumors, but it is also finding a partner with Bayer that is truly committed to cell and gene therapy. They have recently established a new cell and gene therapy unit that is really creating the right type of organization to cover the ability to do different type of partnering. And we are working very closely with them onto the development, as you can see on the next slide, of the first allogenic CAR T that will address mesothelin. And mesothelin is overexpressed in many solid tumors, from advanced mesothelioma to also non-small cell lung cancer, ovarian cancer, pancreatic cancer. And we have already great data from a preclinical work that we presented last year showing the unique design enabling the ability of eradicating the tumors and having functional persistence there. Now, on the next slide, you will see that this is leading also to the possibility to develop other type of allogeneic CAR T, and with here a CD19 type of allogeneic CAR T, which is being developed to be potentially the best in class off-the-shelf allogeneic program for B-cell malignancies. This is supported by recent data coming from an academic team at Memorial Sloan Kettering that has shown last year that this type of EBV CD19 CAR T does indeed lead to a high level of complete remission, up to 83% in patients with advanced B-cell malignancies, with a very long follow-up, nearly 27 months of follow-up. And at the same time, this has led to an acceptable safety with no issue with CRS or neurotoxicity. So we are really working ourselves independently to develop this type of allogeneic EBV CD19 CAR T to become the best in class, hopefully, in the field of lymphoma and other B-cell malignancies. Now, I would like to conclude on my last slide, the next one, yes, this, where we are really at Atara trying to advance rapidly on multiple fronts. And we believe that within the next 24 months, we might very well have the first ever allogeneic T-cell therapy approved, hopefully in the US and in Europe. We also should have, within the next 2 years, have data coming from the randomized controlled trial in progressive MS that has the potential to unlock a great opportunity for patients and for the company in terms of showing, hopefully, disability improvement, as we've shown into the open study. And then on CAR T, we'll have multiple programs with clinical data, both in hematological and in solid tumors. And all that being supported at the time, hopefully, by a commercially validated manufacturing platform. Now, my last slide, the next one, will be really around patients, and thanking you for your attention there. We are here for patients with MS, progressive MS in particular, that are suffering from that devastating disease. We're here for patients with deadly cancer, and we believe that our platform has the potential to address a multiple number of serious disease, from autoimmune to solid tumors and liquid tumors. Thank you very much for your attention.
Thank you very much indeed, Pascal. Fascinating work, and all the best for your continuation there. Our next speaker is Miguel Forte. Now, he is CEO of Biotherapeutics. Biotherapeutics have developed a unique approach to cell therapy products for orthopedics and bone diseases. They're creating a new and unique treatment approach using differentiated bone-forming cells administered via a minimally invasive procedure. To find out more, here's Miguel.
Thanks, David. Thanks, everybody, for being there, and thank you for the invitation. I would like to take you through the exciting work we're doing at Biotherapeutics. First, with the traditional disclaimer, and then a quick company overview. We're a clinical stage company in Brussels, listed in Euronext, with 3 assets, 2 in clinic, 1 in preclinical, and a portfolio behind that, backed by a strong IP and really a technology that harnesses primarily the differentiation of mesenchymal stromal cells into a professional therapeutic target. The product pipeline that we have is depicted here on the slide that you see. In the orthopedics, as David pointed out, our primarily focus, we do have a biologic that were used previously to support our cell therapy approach, which is currently in phase 3 in a study that should deliver data before the end of this year, on the 3rd quarter. And I'll come back to some of the challenges we face in recent times and how we managed to deliver that in the current COVID pandemic situation. Our ALLOP, the mesenchymal stem cell derived cell therapy professional approach to perform bone, is currently being developed in 2 indications: difficult fractures and lumbar-spinal fusion. Both of those studies have been, both of those indications have 1 study completed with data release that I'll review with you in a minute, and is currently on a phase 2B controlled study. We have partnered for China and Asia that asset, and we continue to develop elsewhere and engaging actively with potential partners. You will see that with these assets, we currently showing very relevant clinical data that is providing significant value to unmet medical needs. As Pascal mentioned, we're all doing this for the value that we see delivering to patients, for the fun of the science, but above all, for the value that the patients can take. And we have the opportunity and the knowledge with our MSC platform to go beyond, and we have a series of other targets where we, again, with the principle of differentiating cells to specific therapeutic objective, we are moving into indications in inflammation and others still undisclosed. Now, with the keyboard working, I would like to give you a quick preview of one product, not being cell therapy, but nevertheless showing our focus on providing value to unmet medical need. Knee osteoarthritis, a major pathology, a major condition in present days, but our knee suffers from our daily life. We've been able to add to the potential therapy for these patients a viscous supplement that combines 3 elements, and as shown in an animal model, as you can see in the left-hand side, the opportunity to allow the cartilage to regenerate with the corresponding clinical data on a study finished where we've shown superior value versus an active control 3 and 6 months post-injection. We're currently conducting the follow-up of a fully recruited study that we've been able to do that while the challenges of COVID across Europe and in Hong Kong. And that data, that study should provide data on the 3rd quarter of this year. The exciting opportunity that we're bringing with this product comes to the point that currently there is no available asset that can deliver a regenerative approach to cartilage. So we still address the unmet medical need, and the data there and the prospects significantly represent an opportunity for the patients and the company. Now, if I take you to the cell therapy approach and where we want to really become a preeminent company in terms of professionally therapeutic targets derived from MSCs, is based on the technology shown to you on the slide, where from a bone marrow of a donor, we extract the mesenchymal stromal cells that we differentiate and formulate in large number of doses, cryopreserved. This allogeneic approach, this scaled approach, and this off-the-shelf approach provides an industrial opportunity to deliver the high technology of cell therapy in the unmet medical need of orthopedics, because we have differentiated the cells to become professional bone-forming cells, as David mentioned to you in the beginning. We have seen in preclinical the ability of these cells to double the speed of bone regeneration, and we aim in the studies to document our ability to go from the natural evolution of slow resolution of fractures, shown on the right-hand side by the gray line, to an accelerated resolution of those fractures, all patients getting better faster out of the hospital back to active life, and very importantly, a reduction of the late-stage complications of delayed union and non-union. In fact, in those late-stage complications, we have conducted a study which is finished where we recruited patients with fractures non-healing for more than 3 months, 3 to 7 months. In other words, the criteria of delayed union across Belgium and Germany, and where, as you can see on the right-hand side of the slide, through a minimally invasive procedure, we've placed the cells inside the fracture site, and we're able to kind of reignite the resolution of the bone formation. These cells hit the ground running as bone-forming cells, and really, as you can see also on the picture there, from a non-healing fracture, we were able to get osseous callus there with the corresponding clinical benefit. Safety-wise, it was very well tolerated, an allogeneic approach with no safety concerns, and then an efficacy that was able to show the ability to form bone radiologically, as well as reduction of clinical symptoms. A quick show here in this graph presents to you the bone forming ability and the clinical improvement. So bottom line, in addition to the extensive preclinical data showing this ability to accelerate bone, we have documented already the tolerability and the ability to form bone in patients with fractures. Further to that, we have also been able to document the ability to form bone between vertebral bodies in patients with back conditions in a study that we've recently completed also in Belgium, where we've placed the cells in between the vertebral bodies together with a granules and a cage and assessed the formation of bone that you can see there radiologically in 3 sectors. This was able, through this, we were able to document the ability of the cells in a safe way to produce bone, immobilize the vertebral bodies, and consequently provide a clinical benefit. You can see here, the ability to have full bone formation in the 3 sectors shown in the radiograph before, as you can see there, we had 90% of the patients with that complete fusion and partial fusion on the others, as well as a significant improvement of the clinical condition. So we have clearly documented the tolerability and the ability to form bone. And so we are now engaged on a study for difficult tibial fractures, fractures that are prone for complication and slow resolution. That study has started and is actively recruiting now. Very pleased to see the patients being recruited despite the challenges across Europe in the hospitals. And it's a very simple study where a patient with an accident, a significant tibial fracture, goes to hospital, gets surgery to get the fracture aligned, and then gets randomized into control, i.e. standard current practice, which is expectant approach to the resolution of the fracture, versus an active approach where we, as I've shown in the previous images, place through a needle these professional cells into the site of the fracture, hoping to document again the tolerability and that acceleration of fracture resolution that we saw on the graph, and prevention of late-stage complications. This study is expected to deliver data next year. Now, taking our expertise on differentiating and professionalizing MSCs, we're also going beyond, and we have already in preclinical a product that we developed with targeting the inflammation in the lungs, namely in the context of COVID. And the principle is very simple. From bone marrow or other potential sources, we isolate MSCs and then educate them, differentiate them, or gene modify them to a professional therapeutic target. Again, the concept of ALLOB, of getting professional bone-forming cells, can be extended to other indications, again, using the cell therapy approach, but modified and professionalized, educated to a specific target. Now, all of this, and I would like to bring together the recent advances on the company and what have we been able to achieve. I would like to show to you the focus on recent partnerships, namely highlighting, as Pascal mentioned before, the extensive collaboration that we are engaged. First, in licensing with ALLOB or a couple of companies in China, which validates the technology because of their approach and their experience in cell therapy, and the other company being a commercial company with the interest of taking this asset for commercialization. Further to that, they will do development at their own cost and help us to together generate faster the data required for submission. In addition, we have established a strategic approach with Catalent for a supply agreement in terms of being able to manufacture throughout the world, and very actively on expanding our portfolio and our research. We've engaged with Regenerant for process development and new product development along the lines of I mentioned to you before, as well as other academic collaborations for bone and other indications. Overall, we've been able to achieve multiple objectives, again, with the intention of bringing value to patients through well-documented clinical studies. We have completed the recruitment and expect data on GTA this year. We're actively recruiting for the ALOP study. We're expanding our portfolio, and we're very active on linking with other organizations to further our down-the-road commercial opportunities to bring these products to patients all over the world, as well as expand our research. In reality, we have 3 priorities at Biotherapeutics: focusing on delivering the clinical data and taking the products to the market for the patient's benefit, collaborating with multiple organizations to out-license and in-license to expand the technology, and really, through that, continue the development of new targets, new products, again, and foremost, for the benefit of patients. David, over to you, and thank you to the audience for the listening.
No, thank you to you for taking time out to present to us. A massive thank you to Miguel and Pascal. If you have any questions for either of our speakers, then join us for the Meet the Speaker session, where you can interact with them face to face. This is coming up next on the network track. After the Meet the Speaker session, we have a panel discussion. That's hosted by Akram Biotech on raising the bar on commercial products to have GMP materials in the COVID-19 area. So, hope to see you back here soon.